Many believe learning is simply the act of filing away information, like storing a document in a cabinet. This is a myth. In reality, memory is not a static file. It is a continuous process of reconstruction. Every time you recall a memory, you modify it. Learning is the process of actively updating your internal model of the world. This dynamic system ensures that your brain remains a living, evolving landscape rather than a frozen library. Learning is not a whole brain event. It happens primarily at the synapse, the microscopic junction between two neurons. When a neuron fires, it releases chemicals across this gap, sending a signal to the next cell. This constant activity leads to the core principle of neuroplasticity. Neurons that fire together, wire together. By repeatedly crossing this synaptic gap, the connection strengthens, physically building the hardware of a new memory. Synaptic plasticity is the fundamental biological process that allows our brain to change and learn. It is the ability to adjust the strength of connections between neurons based on their pattern of activity. Long-term potentiation, or LTP, is the process of strengthening synaptic connections. When two neurons communicate frequently, the synapse between them becomes more efficient, essentially cementing the memory trace. In contrast, long-term depression, or LTD, weakens connections that are not frequently used. This synaptic pruning is vital. It removes neural noise, allowing the brain to focus its energy on relevant, high-priority pathways. A memory is not a vague concept. It has a physical address. This is called an engram. Researchers can now tag specific subsets of neurons that store a memory, allowing us to trace them in the hippocampus and cortex. By activating these tagged neurons, we can trigger the entire memory recall process. It is a biological data node in a vast network of connections. Mapping these engrams reveals that learning is the process of physically restructuring the brain's circuitry. For decades, neuroscience focused almost entirely on neurons. But today, we recognize that learning is a collaboration between neurons and their essential partners, the glial cells. First, meet the astrocytes. These star-shaped cells serve as the brain's metabolic engine, delivering the energy and nutrients required for neurons to form new connections during learning. Then, there are the microglia. Acting as the brain's specialized cleanup crew, they prune away weak or unnecessary synapses to clean up neural signals, essentially sharpening the network's efficiency. This constant pruning refines our neural circuits. By removing noise and strengthening relevant pathways, glial cells ensure that the brain remains a fast and efficient information processor. Most regions of the brain stop growing in volume after puberty, but the hippocampus is a remarkable exception. Throughout adulthood, the hippocampus continues to produce new neurons, a process called adult neurogenesis. This process starts with neural stem cells, which can branch into neuronal or glial progenitors, eventually maturing into functional neurons. Why does this matter? These new neurons facilitate pattern separation. This helps the brain distinguish between similar memories, such as where you parked your car today versus where you parked yesterday. Targeted memory reactivation, or TMR, is a revolutionary technique that uses sensory cues during sleep to strengthen specific memories. By playing specific audio cues while a person is in deep sleep, we can trigger the brain to replay and solidify newly learned information. This experimental design proves that sleep is an active period of rehearsal, allowing us to accelerate the consolidation of complex skills. Research confirms that TMR enhances signal-to-noise ratios in neural pathways, making memory formation a recoverable and manageable process. Within 24 hours, our brains discard roughly 70% of new information. This is known as the forgetting curve. Biologically, this happens because synaptic connections that are not reinforced begin to weaken, a process that improves neural efficiency by clearing out noise. To counter this, we utilize spaced repetition. By reviewing information at calculated intervals, we interrupt the decay and solidify the synaptic structure. Physical exercise is not just for the body, it is a chemical trigger for the brain. A session of 20 minutes of moderate to high intensity exercise is the threshold for releasing a vital protein. Hitting the 20-minute mark releases brain-derived neurotrophic factor, or BDNF. Think of BDNF as miracle grow for your neurons, supporting the growth of new connections. This spike in BDNF opens a plasticity window. For the next 60 to 90 minutes, your brain is biologically primed for high-intensity learning. This is the optimal time to tackle your most difficult mental tasks. By aligning your study schedule with your exercise routine, you utilize this chemical window to maximize neural efficiency. Memory is not a static file we just read. It is a creative act of reconstruction. Why is testing yourself superior to simply rereading? Passive study, like rereading, gives an illusion of mastery. However, active recall, forcing your brain to retrieve information, creates a necessary cognitive struggle. This struggle is exactly where neural plasticity happens. Every time you successfully retrieve a memory, you physically update and strengthen the neural pathways associated with it. Rereading is passive, but active recall is a workout for your brain. By constantly testing yourself, you ensure your knowledge model is accurate, robust, and easily retrieved when needed. Every brain processes information uniquely. When we talk about neurodivergence, like ADHD or autism, we are looking at brains that often encode memories through different neural pathways. While standard pathways often follow linear models, divergent brains may utilize complex, parallel, or specialized networks. These aren't defects, they are different architectures for processing data. Research shows that these methods are highly efficient when the learning environment is optimized for the specific neural profile. Success depends on aligning the input structure with the individual's encoding strength. In 2026, AI brain interfaces are redefining personalized education. At the core of this technology are neural digital twins, sophisticated AI models that mirror an individual's unique neural activity patterns. By collecting real-time neural data via advanced EEG sensors, these systems construct a high-fidelity digital twin of the user's brain processes. The AI then simulates how this digital twin reacts to various educational stimuli. This allows systems to predict, with remarkable accuracy, how quickly a learner will master a complex skill, dynamically adjusting the educational pace to match the brain's optimal learning rhythm. By matching educational pacing to your unique neural architecture, we are moving beyond one-size-fits-all learning into an era of high-precision intellectual development. When we experience stress, the HPA axis initiates a rapid response. The hypothalamus triggers the pituitary gland, eventually releasing cortisol into the bloodstream. While short-term cortisol prepares the body for action, chronic stress floods the brain with this hormone. High levels of cortisol effectively inhibit the hippocampus, the brain's primary engine for memory formation. This makes emotional regulation a physiological necessity. By lowering stress, we reduce cortisol, reopening the synaptic pathways required for effective learning and memory storage. Epigenetic priming represents the frontier of learning science. It describes how our biological state makes the brain more receptive to encoding information. Epigenetic markers act like a volume knob for gene expression. Lifestyle factors like sleep, nutrition, and exercise add chemical tags to your DNA. These tags modify how accessible your genes are. A primed brain is one where genetic pathways related to synaptic plasticity are fully accessible, ready to strengthen new connections. Future educational research aims to leverage these markers, identifying the ideal biological states to maximize the efficacy of your learning sessions. Reconsolidation is a critical property of memory. When we recall a memory, it enters a labile or changeable state. This process allows us to update our past experiences with new information. Experimental evidence, such as boundary extension, shows that our brains fill in gaps in memory with expected surroundings. We don't store snapshots, we store blueprints that we rebuild every time. Because recalling a memory involves activation and subsequent storage, the memory is subject to modification. This is why memories can become distorted or strengthened based on how you feel right now. Researchers have discovered that the brain does not just store memories passively. During rest, the brain actively replays experiences to master them. By monitoring neural activity, scientists watched in real time as the brain replayed maze navigation patterns during periods of inactivity. This simulation allows the brain to solidify skills without external input, providing visual proof that learning is a dynamic process of reconstruction. Traditionally, neurodegenerative conditions were viewed as a one-way street of inevitable decline. However, recent medical advances are challenging this narrative. New clinical trials are exploring the use of targeted neural stimulation to reactivate stalled consolidation pathways. By identifying these dormant routes, we can bridge the gap in memory function. This changes our biological understanding. We are moving from a focus on cellular death to the restoration of neural communication, framing cognitive health as a potentially recoverable process. The brain learns best when information is processed through multiple senses simultaneously. A single sense creates only one neural pathway. If that path decays, the memory is lost. However, engaging additional senses adds redundant routes. This creates neural crosstalk, where sensory information reinforces the same core memory trace from different directions. By distributing the memory across these multiple pathways, the information becomes much more resistant to forgetting. Learning is an energy-intensive process. Glucose, the brain's primary fuel source, is essential to sustain the high metabolic cost of creating new synaptic connections. Building new neural pathways requires building proteins and maintaining cellular activity, processes that drain our energy reserves. Rest and sleep are vital. They allow the brain to replenish metabolic resources and consolidate the connections forged during high-intensity learning. To prevent forgetting, we use spaced repetition. This strategy aligns our learning intervals with the brain's biological need for protein synthesis during synaptic consolidation. By reviewing information just as recall begins to fade, we trigger reconsolidation. Each interval allows protein synthesis to reinforce the neural pathways. Think of these peaks as protein synthesis cycles. These intervals give your hippocampus the time needed to effectively teach the neocortex. Apply this strategy. Review at increasing intervals to build robust permanent memories.